Hi, it's Abby here again doing my second vlog. We are now halfway through the academic year and I've already completed one of my 3 modules. As I mentioned in my last video, my main project is investigating biocontrol agents for downy mildew and whether they affect the pea plant's microbiome. Today I thought I could give a bit more insight into why I am doing this project. Peas are grown worldwide, both for human consumption and to produce animal feed. In 2018, the field pea and broad bean production totaled 4.4 million tonnes. But these peas can be affected by a variety of pathogens, and one of the most damaging plant diseases is downy mildew, and this is caused by Peronospora vitae. When a plant is severely infected, the crop yield can be reduced by up to 70%. The downy mildew can affect the plant at any stage in its life cycle. A seedling infection will lead to stunted growth and a plant with yellow blotches on the leaves, whilst a pod infection will not create noticeable change to the plant, but the pods will become discoloured and bitter to eat. In the UK, there is no approved fungicide for the treatment of downy mildew. This is due to safety concerns regarding the only treatment that was available. Therefore, scientists are looking for effective but safer treatments. And one such treatment is the use of microbial biological control agents, which I briefly mentioned last time. So currently there are no known microbial biological control agents approved to suppress downy mildew. However, there is work screening some. The concern for us is that these microbial biological control agents aren't targeted to a specific pathogen. Instead, they work by producing toxic compounds like hydrogen cyanide, or producing non-specific antimicrobial compounds. This means that whilst they may kill the pathogen, they might also damage the plant's natural microbiome. And we know that the plant's natural microbiome is really beneficial because it can increase a plant's resistance to abiotic stresses such as drought, but also reduce a plant's susceptibility to other pathogens. So if our microbial biological control agents damage this microbiome, then they may make the plant more susceptible to other diseases and stresses, meaning we haven't fixed the problem, we've just changed it. This is why my research looking at these potential agents and how they affect the natural microbiome is important. I'm conducting this research by extracting the DNA from the plant's microbiome and conducting metagenomic work to determine whether the relative abundance of certain species in the microbiome changes when infected with downy mildew, and then when treated with the microbial biological control agents. I have recently sent my isolated DNA for sequencing, so I'm very excited because in the next few weeks I should get my raw data back, and I cannot wait to start the metagenomic work because that's something I've done very little of up to this point in my degree.